Hello, this is video number 10 for the immune system. This is the last video for this chapter, and I have just a couple topics to cover. So those are shown on the slide. So what is the basis for immune memory? Well, I want to mention that a little bit. The second topic for this video is active versus passive immunity. All right, so for immune memory, just basic description first of all. Your adaptive immunity has memory, which means that if you're exposed to a given pathogen more than once, the second or any subsequent exposure is going to be more vigorous and more rapid than the first exposure. So memory means that you mount a more effective defense against a pathogen because your immune has some kind of memory. It knows that it's seen this pathogen before. So what's the basis for that? So let's describe what happens the first time you encounter a pathogen or a foreign antigen. And this is called the primary response. Okay, the first time that you encounter a foreign antigen, this foreign antigen can activate B cells, helper T cells, and cytotoxic T cells, as long as their antigen receptor can bind to this antigen. Okay, so this will apply to all three of those types of lymphocytes. If activation occurs, there will be direct contact between the lymphocyte and the antigen, binding to the antigen. Okay? On your first exposure, there aren't going to be very many cells in your body which will have an antigen receptor capable of recognizing and binding to this foreign antigen. Remarkably, you have so many lymphocytes that almost any foreign antigen will have some lymphocyte that can recognize it in your body. But if you've never seen it before, there aren't very many. Not only that, but there might be a significant lag time between the time that the antigen enters the body and it encounters a cell that can bind to it. So, if it gets into your skin, it might circulate through the lymph, go through a few lymph nodes, and even get into the bloodstream, get to the spleen, before finally it might be recognized by a lymphocyte and activate that lymphocyte. Okay, so just because you don't have very many cells that can recognize this antigen, the first time you're exposed to it, it's going to take a while before it's going to activate anything. When lymphocytes are activated, though, eventually one part of that activation results in formation of memory cells. Okay? Then in the secondary response, the second or any subsequent time you're exposed to this pathogen, you already have now a bunch of memory cells, many more that can recognize this pathogen in your body than the first time you were exposed to it. So that's going to shorten the time for a cell that can bind to this antigen to bind it and be activated and elicit a response to it. Okay? In a secondary exposure, typically the pathogen, bacterium or virus, or any other type of pathogen for that matter, will not be able to reproduce enough to cause any kind of symptoms to develop. That's not usually the case for a primary exposure. Okay? Primary response is slow enough that the disease runs its course. And a secondary response, usually very few, if any, symptoms are seen. Good example of that is if you were already exposed to a virus, flu virus. Flu is not a good example because flu changes a lot, but measles, I guess, is a good one, right? So if you're exposed to measles in childhood and you've had that disease, it's run its course, that's your primary response. And if you are exposed to it a second time, you won't develop the symptoms of measles because you'll generate a secondary response. And this whole difference between primary and secondary response is the basis for vaccines. Vaccines are an injection of a part of a pathogen which gives you exposure to that pathogen with a primary exposure without having to develop the symptoms of being infected by the whole pathogen. All right, so this slide shows a humoral response. Just because antibody concentration is easy to measure, but it would be similar for a T cell, helper cell, or a cytotoxic cell as well. It's illustrating what happens in a primary response, first exposure, versus a secondary response, second exposure. And what's being measured here are antibody concentrations. Titer is another name for concentration. And it's measuring two different types of antibodies. We're going to focus on IgG, immunoglobulin G. Okay, and on the bottom is days from first exposure. So 0 is time of exposure, and then this is running through 3-4 weeks after exposure. Left panel is the primary response. Right panel is the secondary response. Okay, so one thing you should notice is there's a lag phase between exposure times 0 and when you start to see an increase in antibody concentration, IgG here. So for IgG, that's about a week after the first exposure for the primary response. For the secondary response, the lag period is much shorter, so maybe a day even before you start to see an increase in this IgG. So this is indicating that there is activation of B cells, and the B cells are undergoing clonal expansion, and clonal expansion is going to generate plasma cells, the differentiation, and the plasma cells are going to secrete antibodies, and those antibodies are going to start appearing in the bloodstream. This is measuring blood serum antibody concentration. Okay, that takes a week for all that to happen from the first exposure. It takes only a day or so for the second or any subsequent exposure. The other thing you should notice is the concentration of antibodies that are produced. First exposure, they rise, they peak, and then they drop. So they peak, they start rising at about 1 week. They peak around 2 weeks or shortly after 2 weeks. And then they drop. By 4 weeks, they're not much higher than they originally were. Okay? For a secondary response, they rise dramatically and very significantly, much higher than from the primary response. And not only that, they peak and they start to decline, but it's a very shallow, slow decline. And after 4 weeks, there's still a huge amount of this specific antibody circulating in the bloodstream. Okay? So the conclusion from this, and this is for the humoral response only, but this will also apply to helper T cell activation and response and cytotoxic T cell activation and response. Primary response is much slower, a longer lag period, and it produces less of an increase in antibodies. And that increase goes away more quickly than in a secondary response. Shorter lag time, dramatically higher increase concentration of antibodies, and it doesn't go away even after 4 weeks in time. Okay? So this is memory. Second time or subsequent time you're exposed, your immune system has memory cells socked away in lymphatic organs, making it much more likely that this pathogen is going to bump into one of them and activate them quickly. Whereas it takes a while to find a cell that recognizes the pathogen in the primary response. All right, so this slide is just summarizing basically what I said on the previous slide. In a primary response, there's a long lag or latent phase, which is the time it takes for a pathogen to bump into lymphocyte that has a receptor that can bind to it. And then it takes a while for all of these processes, binding, activation, clonal expansion or proliferation, and differentiation to plasma cells to take place. All right. In a secondary response, the lag period is much shorter, and that's just because there are so many memory cells with the same antigen receptor present in the body. And the antibody production is much higher and lasts for longer. All right, second topic and the last topic that I'll cover in this chapter has to do with active versus passive immunity. So what is the difference between active immunity and passive immunity? So we get immunity either humoral or cell-mediated. Active immunity results from direct exposure to a antigen or a pathogen. And from this direct exposure, lymphocytes will be activated and they will generate memory cells. Okay, that's active immunity. This direct exposure to an antigen can be through infection or through vaccination. So infection would be a natural active immunity, and a vaccination would be an artificial active immunity. Okay, how does that differ from passive immunity? Passive immunity results not from direct exposure of the individual to a pathogen or foreign antigen, but by obtaining antibodies, typically from somebody else. So how could that ever occur? Well, it can occur naturally, for example, during pregnancy. So a mother carrying a fetus, their bloodstreams are there's connecting through the umbilicus and the placenta, and antibodies from the mother can cross into the fetal bloodstream. So even though the fetus is not directly exposed to an antigen, it can get antibodies from the mother that will protect it. And so those antibodies will circulate in the fetal circulation, and even in the newborn's circulation. They'll last for a while, weeks or so, maybe a couple months. Okay? Also, breast milk passes some maternal antibodies. So that passive immunity from the mother can last even longer after birth if the infant is breastfed. So that's an example of a natural passive immunity in which a fetus gets antibodies from the mother which can protect the fetus and the infant. An example of artificial passive immunity is if you get an injection of antibodies from somebody who's been exposed to a pathogen or an antigen. An example of that is right now, actually, people that have been exposed to the COVID-19 virus can have antibodies from their plasma donated and used, injected into, people can get a transfusion of plasma from somebody who's suffered and recovered from COVID-19 into someone who's currently suffering from somebody with the infection. So they will get the benefit of antibodies from someone who was just exposed and recovered to help battle their active infection. Another example of artificial passive immunity is the venom from certain snake bites. The antivenom is antibodies that were developed by injection of venom into animals. And those antibodies are the antidote to some, not all, but some snake venom cases. Okay, for passive immunity, one of the key differences, you're not directly exposed to the pathogen or the antigen, and so you do not develop your own memory cells. And so you don't develop memory to that particular pathogen. The other difference is the immunity that you have is only temporary. It won't last. It'll last only as long as the antibodies will last in your circulation. Well, that ends video number 10, and that ends the coverage for the immune system. So I'll close it here, and we'll see you later.